very much for the opportunity today. My name is Emma Palmer. I'm a clinical geneticist in Sydney, Australia, and today I'm going to try and provide some guidance on your genetic test report. So firstly, acknowledging that the journey to get a diagnosis of SCN2A condition in your child is often a very long and difficult one. Firstly, your doctor's understanding that your child's condition could be genetic, then decision making about whether to have genetic testing or not, accessing genetic testing, and then finally getting a report. But what does it actually mean? Because genetic tests tend to be written in a little bit of very scientific language, and they can be quite daunting to understand because our genetics is very complicated. We have a very long DNA code, and we all vary in our DNA code. So sifting through what our genetic changes actually mean something and are important for our health or our child's health, and which ones are just benign variations can be quite difficult. But your test report should include these things. So firstly, very clearly that it is your child that was tested, and also an indication of whether that was just a test on your child by themselves or whether you as parents were included, which can be quite a powerful way of doing the test. What type of test was done? Was it just a single gene? Was SCN2A the only gene looked at, which is quite rare these days? Or more likely, was there a panel of genetic epilepsy or intellectual disability autism genes that were looked at? An exome, the coding regions of our DNA code, or indeed the whole DNA code, the genome. The laboratory should very clearly state whether the genetic change found in your child is a clear-cut answer. So is this what we call a pathogenic or even a likely pathogenic finding, which means that there's a genetic change in SCN2A that is consistent with a diagnosis of SCN2A condition? Or is there no genetic change found, or indeed a variant in the gene, but the laboratory does not think this is the explanation, what we call benign? Or, which is the most difficult sort of report often at all, is a VU, so a variant of uncertain significance, which means that more work is going to need to be done to try and understand, is this the explanation for your child's condition or not? So every type of report should include information on not only what the change in the DNA, the genetic code was, but also how that is predicted to affect the protein that is made by the SCN2A gene, so the NAV1.2 channel. And there are really three types of main classes of genetic changes that the report may include. As mentioned before, a benign variant, so that means there's a change somewhere along the genetic code, but it's predicted the protein would still work absolutely fine. So that's not an explanation for your child's condition. Or a group of conditions which reduce the amount of ion channel that's made, and there's a range of genetic changes that can cause these. There may be small deletions, missing bits of the gene. There may be a change in the splicing where the little coding bits of the protein are put together, and if that's scrambled, often there's a lack or a reduction in the ion channel, and changes in the DNA code that make the from the gene to the protein, the message is much shorter, and again it's a scrambled message and less protein is made. Or missense variants. A missense variance is where one amino acid, which is the building block of the protein, is changed. And that can have a range of impacts. So far we know that this class of changes is more likely in individuals who have got a primary diagnosis of an intellectual disability or autism, but they may still have seizures, as opposed to missense variants, which are often linked to a more severe or more prominent seizure phenotype and less likely autism. But neurodevelopmental delay may or may not occur in this group of conditions. So for missense variants, what's really important is which bit of the ion channel is affected. So there's a blog post here where you can visit epilepsygenetics.net that talks about the SCN2A protein and gives an idea that there are some areas of the protein where no genetic changes have been found. There's no coloured blobs in any individual. So what that's telling us is those are probably parts of the protein where it doesn't matter if there's a genetic variant. However, there are other areas where you can see lots of little blobs, and the blobs are where there's been children found with SCN2A related condition. And if there's alterations in the amino acids, those building blocks in these critical regions, that's very likely to result in a neurological or neurodevelopmental changes for your child. And there are various tools that can be used to actually try and plot. If you know the genetic code or you know the protein change, then you can actually plot and find out where in the ion channel those changes have been found. So that's something that your geneticist should be able to do from the report, and that can be very helpful information. So often the laboratory needs to make a judgment whether a missense change, that's one of those changes in the amino acids, is the cause or not. Sometimes they can be very clear cut, like I said, giving a pathogenic result, or they can be more hesitant, and it's in this variant of uncertain significance, or indeed they say it's very likely to be benign, not the cause. And these are the sort of things that are weighed up in that consideration. So firstly, if we see the same genetic change recurrently in other children with SCN2A condition, well, that's good evidence that's likely to be actually the cause in your child. Conversely, on the other hand, if we see it in lots of people in the general population who've never had seizures or neurodevelopmental problems in their life, then probably that's just one of those benign variations that doesn't matter at all, and it's not the explanation. Like I highlighted before, is the amino acid that's changed in what we understand right now to be an important critical part of the ion channel. And this isn't a complete information yet. We're still reading and learning the book about SCN2A, so we may have more information in a few years' time if we don't fully understand things now. And then what kind of altogether evidence is there that this amino acid affects the ion channel structure or function? Has anyone done functional studies in the laboratory, seeing how the ion channel is changed? Is it a part of the channel that is very, what we call evolutionary conserved? It's present in all different types of animals that have this type of ion channel. And then also, do we see it in other people in the family who do have this similar symptoms or who don't? So if we see it in lots of people with similar symptoms, that's good evidence towards this being an explanation. But on the other hand, if we see it in people who have never had neurological symptoms, learning difficulties, or indeed seizures, then that may be evidence against this being the likely explanation. More investigations need to be done. So I'm coming to an end of my talk, but I just wanted to highlight some resources that you could look at for more information. The fabulous webinars and podcasts that are on SCN2A Australia, highly recommend those. Catherine Howell has put together an information sheet, which I helped a little bit with in the genetics section, and that's available on Human Disease Genes. So that's humandiseasegenes.nl/SCN2A/. Also, if you're more interested in trying to decode the genetic terms in your child's report, to understand more about different types of variants, a good resource is the Centre for Genetics Education. It's got lots of fact sheets, for example, on missense changes, on inheritance patterns. And we've tried to help by producing some information on our website, which is pennewsouthwales.com.au. If you go here, you'll see this new genetic information for families. And these are resources that we've co-developed with families to try and understand and answer some of the questions that families have about genetic epilepsy, inheritance, genetic testing, and also what to expect in a clinical genetics appointment. And also we link out to more information and resources. So thank you very much for your time. Also feel free to reach out to myself or for your doctors if they were wanting to link up. That's my email address, and we also have a website which gives a little bit more of information about our multidisciplinary research team. So thanks for your time, and I hope you do feel very, very welcome to reach out to me if you have questions.